Yesterday, the phenotyping group followed on from the discussion that had occurred earlier in the week, and probably to some extent may have been a follow-on from what Kurt's discussion was. A lot of it seemed to center around development of more indicator traits for hard-to-measure component traits. How do we get things to, in essence, increase inheritability? But to do that, you may have to measure a lot of things that we otherwise have not been doing, and one that was talked about quite a bit was images, whether that was real-time movies, thermoimaging, FTICR, you name it, numerous different portions for it. In order to do that, back up. For the phenotypes for those components, it was really over about anything and everything that people could think of. It varied with the individual who was there as to what traits that they put as the highest. So some of these will be species specific, some of them would go across species like feed efficiency or disease resistance. But the needs in order to try and help facilitate this phenotyping was need for some standardization of different types of methods, because some of these things could be measured in numerous different ways, but have people collected enough information with that in order to know how to integrate things across. There was hope that with some of these new things that we could actually make dramatic leaps forward. It also was discussed about how the phenotyping would benefit numerous different things, not just precision selection, but also potentially precision management, and some of these things will go hand in hand. In terms of two-year goals, one that was considered probably more low-hanging fruit is how to try to get access to pre-existing data. It's already collected by industry and other places, but may not necessarily be available for folks. That people need to talk directly with industry in order to try and do that. And this goes back to a crowdsourcing discussion that had occurred of how to try and collect it as well. Some of this we do realize it depends upon industry and what are the forms that it is, the format, the organization of the industry, and other portions would impact that. There's probably a need for development of new technologies that would occur in the next 2 years for phenotypes, whether that's a wearable tape or something else to collect something at a cheaper price than what it is that we would do today. In order to do that, we probably have to engage with engineers and software developers to a greater extent in order to facilitate that. It was also believed that some of the standards and protocols for omics type data in order to facilitate this component type of analysis could occur within the next 2 years in order to try to integrate data sets across. Also need to train folks and how to collect and utilize some of the technologies that exist today, as well as those that would be oncoming, additional training for data analytics and visualization. 5-year longer term desire would be from omics type things. Are there things that you could actually collect in real time, that you don't have this large lag from when an experiment was run before, say, gene expression would come online? Is there a way to focus on the translation of the discovery that occurred in the last 2 years to where in 5 years it actually has translated into new applications within industry, and that they would probably be specific for each of the different species? There's probably going to be a need within that 5 years to have training for industry, so back to the extension outreach component, in order to allow industry to fully utilize the new technologies for phenotype collection. Are there things in which I forgot? No? Okay. Now who's next?
We had some overlapping discussion and on our board, our group was the host pathogen interactions with myself and Hans Chen. And we also talked about, as Jerry mentioned, the return on investment, and we thought that the return on investment for a lot of the studies that had already occurred for host pathogen interactions were, as he mentioned, substantial, and that this might be something that would be worth bringing up to the legislators or whoever, and how that could impact animal agriculture and food safety in the United States. So here are some of the topics that we discussed. First, we kind of broke it down into host and pathogen, and then the environment, and then some kind of overall outcomes. And for the host, we discussed that the heritability estimates for the susceptibility or resistance or tolerance, depending upon what your goal was, for the disease was important to identify the opportunities for genetic change over time. And those that had higher heritabilities, there's probably greater opportunities, and they might have a larger focus on those particular diseases than those that had particularly low heritabilities. We also thought it was important to identify the pleiotropic effects and to identify how the correlations between disease susceptibility, if we made genomic selection against a particular disease or tolerance, how that might affect production traits, and or it might have effects on other diseases as far as the resistance or susceptibility or tolerance to other diseases as well, and that those needed to be vetted out. We also talked about the identification of loci associated with disease, may be further evaluated using comparative genomics to exploit the information obtained in other species. And we thought to do that effectively, we needed a complete genome assembly and annotation of the functional regulatory elements in the different species, and that this would then facilitate information across species, and we would get a bigger bang for our buck, so to speak. We also talked to make genomic selection available to the livestock sectors. We had to agree upon some common definitions of disease and how we were going to measure those so that they were consistent across studies, so that the information would be additive across studies instead of disjointed. And that will have to involve stakeholders and industry so that we can agree upon things that are easy to measure and that we can then use for selection. We also talked about a comprehensive understanding of the immune response, including innate immunity, and tools that facilitate the assessment of the immune response would assist researchers in selecting animals that have superior health potential. When we move to the host, as with the host, the complete genome assembly and annotation of the different variants and pathogens would be important so that we could categorize and identify the host pathogen interactions. And we also discussed that it's important to have inexpensive tools that could be used so that we could do diagnostics of which pathogens were present, especially in multi-pathogen diseases, so that we would be able to actually target the genomic selection loci that were important for specific pathogens, because they may vary, and so the selection also might vary across the United States. So in keeping with that, we also recognize that we need to identify the environment, because that is going to have some modulation of the pathogens, and that the host pathogen interaction to cause disease is reliant on the environment. And so with this in mind, we thought it was important to study the microbiome, or the microenvironment that was important for the pathogens, and understand how the hosts can withstand infection and how this may be altered by vaccines, probiotics, feed additives, or feed alternatives, and epigenetics. And so then kind of on a other aspects category, we talked about some things that might be helpful in accomplishing those goals. And one of those was software that aids comparative genomic studies, perhaps visualization software applications that assist with recording phenotypes in a seamless and time-efficient manner so that people will actually record the data in the industry. And that way it would facilitate adoption by the industry and researchers alike for extending the research studies. Also, we thought that genomic selection will be integrated with precision farming to increase efficiencies and profitability, and to take it a step further, that perhaps the development of a national database that records diagnostic results on pathogens across the U.S. We have these national labs that do the diagnostic testing, but they're not keeping that information. They're not having a repository of the samples so that resource could be mined. And we thought that it may be helpful to start to develop that resource on a national level so that we could start to key in on how things differ across the United States as far as the environment and how we need to tailor selection accordingly. So the relevance of the study of host pathogens is negative impact on animal welfare, and we wanted to recognize that animal welfare is a very important consideration, and that it is also very detrimental to animal performance and production. We thought that as consumers become more concerned with antimicrobial use, the use of alternative means such as genomic selection to improve human health and animal health was very important and is certainly attainable. So we also considered that partnering with other federal agencies such as agencies that are involved with bioterrorism, one way that the United States could be at risk would be if they provided pathogens that were detrimental to our human or animal food resources. And so we thought that we may be able to partner with some other federal agencies to secure funding that might be addressed in that way. So we thought reducing disease through selection will have a significant financial impact, and that our return on investment would be high, and that those might be highlighted for the benefit of funding opportunities for animal agriculture. Anybody have any other questions or input that were in the session that I might have missed?
When you say tailor selection, is that any reference to
What his comment was tailoring selection, was it in reference to Jerry? I'll let you answer that, Jerry.
We definitely see some common themes across these topics. I wanted to ask you a little bit about quantifying the impact of this work. So, did you talk about specific things that you could try to quantify? I mean, we can look at the research funding. That's kind of easy, but to get the impact on the other side is a challenge.
So we used one example in the CAP grant. We did some economic studies to identify if we had. What I mentioned in the session was if you have the heritability estimates, you could use that as a phenotypic accuracy, and then you could go through and say, okay, if genomic selection cost you this much and we made this much progress based on what your heritability is per year, what would that mean in reduced loss, either in production or mortality or morbidity, however you want to frame that, and then translate that into a dollar amount. So we did some of that in the BRD grant, but I think that isn't really that hard to do, and that people could do that, and you could then sum those across some of these projects, and you could say, you put this much in, and we're going to return over $100 million in a year. So that's a pretty good return. Can I make a comment on that? Yeah.
I know I've been at discussions with Peter Johnson with NIFA, and they one of the things for impact and cost of disease is not well documented for many, many diseases. PRRS is the porcine reproductive and respiratory syndrome is one in which we have good documentation that has been recently updated. And so this type of economic information is actually important, and particularly in species that, in other species besides the cattle and pigs.
I think that if people turn their attention in that direction, if they could document that, a lot of the studies could probably come up with figures on that would be relevant. that would be very helpful for you, that when you're saying, you know, look at how this is affecting our national security for our food resources and our animal and our human health, as far as, you know, we're being able to address disease in other ways besides just antimicrobials, I think that would gain traction.
And for the foreign animal disease, you know, just some information of, should we have something, I'm sure DHS has some of that things, so, you know, there's work on African swine fever, but foot and mouth disease are big issues that, you know, haven't, you know.
Thank you. Derek?
So I want to give the report on the microbiome and metagenomics group. So despite the fact that many of the people in my group were rather quite boisterous, we had a very straightforward and candid discussion. But we first wanted to start out with the definition of microbiome and metagenomics, because they've often been hot topics within the research community, but without clear definition. And much of that has to do with their complexity and their specificity in certain studies. So we came to the conclusion that the microbiome, particularly in reference to how it will be used in agricultural systems, needs to be incredibly inclusive, including species that are from the bacteria, fungi, protists, and including viruses as well, because wherever there are bacteria and archaea, there's likely to be phage and virus. So it needs to be all-inclusive of not only the constitutive members of single-celled organisms that live within and around the animal, but also the environment that an animal is coming in contact with, because that can determine disease incidence and other production phenotypes that may be beneficial for producers. And we then sought to define metagenomics, and I have to give good credit to Tim. He summarized it in a single sentence and said basically it's all the DNA in the system. So I think that that is probably the best way to look at it. So all the genomic DNA that's rotating. In the case of some viruses, we may need to include RNA as well, given their genetic material. So much of our group, the reason why it was so straightforward, I think, was because there hasn't been much done in terms of establishing baseline resources for metagenomics and the microbiome in animal production systems. So a lot of the effort in the coming years needs to be focused on the generation of those resources. I'd like to think of all of our systems as being on a timeline. So in many cases, we follow what the well-funded species do, and we're able to play catch-up. In terms of the microbiome, we don't even have a reference genome. We have very limited tools and low resolution in which to study it. So a lot of effort needs to be made in order to interrogate these systems and compare the variability. So, just going through my notes, sorry. What we'd like to do is to propose differences in sampling methods to accommodate higher resolution characterization of the microbiome in animal production systems, and to also generate references for those microbes. This is not going to be something where we can set clear milestones. I think it's an iterative process that we need to constantly re-engage. We'll never have the complete picture, so this is something that will give all of us good job security in the future. So it needs to be iterative, and it needs to have standards and guidelines in order to progress. We also need to establish methods to take longitudinal surveys as well. A lot of people have done single samples and have concluded, oh yes, the microbiome is involved. But we know from experience that microbial diversity can vary based on time point, condition, and other sorts of criteria. So there needs to be some way that we can take longitudinal samples, preferably efficiently and cost effectively, in order to survey this. And the eventual goal is to try to take our information of the microbiome and tie that to production phenotypes or disease incidence. I think that's the holy grail of this survey. After we establish the baseline, then we can start to interrogate this a little bit more readily. And eventually, we want to be able to be the masters of the system and to be able to perturb it in order to gain productive advantage. In some systems, this may be very difficult, particularly as some species form a symbiosis with particular microbes, and it's very difficult to challenge that and to change it. And then finally, we want to take all this information and link it together so that we can start making higher order inferences to model the system, find out what inputs lead to what outcomes or outputs. And I think that's something that will be the eventual goal. And also to study symbiosis, to characterize if there are commensal communities that form a ready symbiosis with the animal, and if there is a state of symbiosis and dysbiosis on an animal-to-animal basis. So that's the general summary. Anyone from our group want to add anything else? I said you were a boisterous group.
I just have a comment.
Yes.
You talked about a lot of standards. You talked about a lot of standards.
Mm.
So there is a group that's led by the NIST. They have a monthly conference calls around metagenomics and then microbiome standards. So, anyone who is working in the microbiome community, so it's a, they have a Zoom conference call. I think I would encourage them to participate in that and then see what's the, they're coming up with the global standards across all species. So just be involved in that effort could be helpful.
So I think that would be an excellent idea. And plus, I think that the animal community needs to engage with that just to ensure that we're not having something that's more, say, human standard or specific. So I do think that engagement with NIST would be very useful. Anything else? Okay.
So our group was the Genome Tools and Resources, and before I forget to say it, because of Holly's discussion of quantitating impact, sorry, I got Adam Filippi's call. Bob mentioned a way of potentially quantifying impact would be to do number of genotyping arrays by zip code. Did I get that right, Bob? Oh, or Jerry. And that might be a way of saying to particular congressional districts, look how much impact we're having in your neck of the woods type of thing. So in terms of tools, as with Kurt's group in the selection thing, the thing is that not all species are the same right now. And because of the selection of people that are here today, we tended to talk a lot about cattle all the time. Sometimes some pigs, but there's some short shrift to some of the other species. So we broke it down into, well, there's the ones that haven't even got started on genomic tools, and then there's the ones that have some advanced genomic tools, and they have to be considered a little bit separately. And for the ones that don't already have it, obviously you have to have a reference genome. So anybody proposing to do genomics and doesn't have a reference genome, you need to be told, nope, you have to have a reference genome. So there was some discussion about at what quality do you need to have, and pretty much consensus position was, given that the tools that are available now, it should be discouraged to do these crappy short read hack job genomes because you're going to end up having to redo them, so it's somewhat of a wasted resource. And the cost of doing the higher quality ones is much better. And then there was a discussion of, well, how good a quality is high quality? And I proposed that it just mean that you have chromosome level scaffolds would be a good measure, because we couldn't agree on an N50 or something. That depends on the species and things like that. But if you can get to chromosome level scaffolds, which is quite doable for pretty much anything. Some of the plants have a harder time, but I think all the animal species aren't going to be that difficult to get to. So, but there's the ones that already have a genomic reference sequence. The discussion was around, is it valuable to have more than one reference sequence? For example, we've back to cattle again, but our reference has been a Hereford animal, and we're recently developed a Brahman and an Angus genome assemblies too, and so are those going to tell us anything that we couldn't have got just by short read resequencing of genomes? And I think we think there will be, but I'd like to see that demonstrated before we decide that that would be a useful paradigm for every species out there. In terms of reference genome, just because you know all the sequence of bases in a row and you know to high quality how they all go together and where they lay on chromosomes, that's not actually useful until you've annotated it. So that brings all the FANG activities into the play, and tried to decide what the standards should be there. We decided a minimum standard would be that at least the animal that you use to create the genome sequence should have RNA annotation available for that animal. Of course, that's only one developmental stage, one time point, one condition, and it would be encouraged to try and enhance that by doing other animals and conditions, etc. But that the minimum would be at least to enough RNA-seq level that you could create a ref-seq in NCBI, for example, for the species to avoid, and that should be encouraged as part of the genome assembly thing, because for a $500,000 grant now, you should be able to do all that. We talked about moving on to the next generation of genotyping platforms that needed to take into account more than just SNPs, and it would be ideal if a single platform could do not just SNPs but also CNVs and structural variants, should that sort of technology become available. In light of John Quackenbush's talk, we talked some about the EQTL and could we create an EQTL network resource population, but after much discussion decided that probably those needed to be, because as John pointed out, those networks there's not a network. So we could create a network, but it may not apply in the situations that you need to. So it would have to probably be relegated to people who may or may not have the capacity to do it, but that the individual experiments probably had to do their own EQTL resource. There was discussion about whether we could have common use resource populations. I would say we were very split on that idea. It hasn't worked well in the past to try and create those because a single resource is probably not going to help everybody out. We're pretty sensitive at the U.S. Meat Animal Research Center because since I've been there since 1992, there's been these waves, well, you guys are going to be the resource for the whole world and you're just going to give everybody your phenotypes and DNA. But that model doesn't work very well because nobody wants to be a service scientist phenotyping for somebody else. But that's still a public resource, and so once we publish, you know, that sort of stuff has to be available, and up to and maybe including tissue. In terms of cell lines, Hans mentioned that cell lines could be useful, especially if you're doing genome editing. We didn't have a lot of time, you know, we were kind of crunched there for the last groups, so we didn't flesh out that discussion very much. Cell lines aren't that hard to make, so I'm not sure whether a common resource is important unless there's some sort of germplasm that really needs to be captured. Each of the species should be strongly encouraged to have a HapMap resource. So, for example, cattle have a set of samples for which there's lots of DNA so that you can ground truth SNP calling algorithms and things like that, and that's proved to be really important to have. So it would be good to build those kind of panels for other species, and also a HapMap. And I'm sorry, we got to the end and went back to annotation. That is, Chris Tuggle suggested that maybe we should look for higher order or higher level annotation like chromatin interaction maps and things like that. Again, the problem is there may not be a chromatin interaction map. Those are also somewhat fluid and may change over time and or breed and or condition. So we didn't actually have time to get to what's a 2 and 5 and 10 year thing in the group. So we'll have to try and work on that as we move forward. Did I miss any major points, anybody?
Okay, thanks, Tim. Janet, are you ready?
Sure, I guess. Well, we had a lively discussion and we didn't always agree on everything. So I'm going to say what I captured in my notes, and then I'm sure others will chime in on something. So we have, of course, the issue because we were trying to look at multiple species. The problem we have is the different species have different success rates for cryopreservation. It's generally poor for the poultry species, and so we need a lot of work in that area. With the chickens and all the avian species, the semen cryopreservation is very variable, different from different lines. But there is some success at that. However, when we just freeze semen, of course, we're missing the mitochondrial genomes and we're missing the female W chromosome. So we're missing a really important parts of the genome. There's a lot of concern that we've had a lot of loss of variation over the last few years. There have been many research lines that have been gone, specialized populations that have been lost. And there's certainly more losses upcoming. And my point is that if we're going to be doing genome editing, those were the sources of variation that could perhaps tell us which changes we should be making. These populations in many cases have been poorly phenotyped, so we don't know what the causative mutations are, but they are a resource to give us that information. We had to talk about doing some biobanking, and this is really critical to do it before we lose much more of our variation. But then the question is, well, how many samples, what types of tissues from which lines, populations, breeds, resources do we do this biobanking? We also discussed this biobanking probably should be more at the population level versus the individual level. Signatures of selection. We've got some very interesting breeds, heritage breeds, some research lines, inbred lines. We've got lines that are divergently selected. What is the difference between the genomes of those birds versus the highly selected, productive birds that are used for animals, that are used for meat, milk, egg production? I already mentioned this, that these variants are sources of functional variation that as a resource for genome editing. Measuring genetic diversity, in a lot of cases we have no idea what kind of genetic diversity we still have remaining. And there's really no mechanisms for following up on that. Also, HAP maps, that was just mentioned in the last one, and there are no HAP maps for chickens, and I'm pretty sure that there's no HAP maps for any of the other poultry species as well. There are HAP maps for cattle and for sheep. I think Noel said that sheep have them. Now, one thing I've noticed in our discussions is we didn't touch aquaculture at all. Sorry, Caird. You weren't there. Okay, so I don't know that much about aquaculture other than the fact that I like to eat some fish. But I think that's a really important piece that needs to be in this whole picture of animal genomes, variable animal genomes. So those were my notes that I had. I'm sure somebody else is going to have something else to add, so here you go. Anybody else? Julie, you want to say anything? No.
Noel.
So I think the differences between the species really is important here, with the avian species unable to actually recreate lines from the germplasm. And so one of the things is some research in that area that could allow those types of things. And then again, this idea that there's the species that have and the species that don't have. As Janet said, the HapMap would be a really good foundation for starting to look at genetic variation across these different species, which would then lead into which samples would be best for conservation. So just reiterating what Janet already said.
Did your group consider either PGCs or embryonic stem cells as a material to cryopreserve, since those can be regenerated in a fairly straightforward way now in both chicken and quail? In theory, you could expand that to all the birds. I mean, we're not stuck with eggs and sperm today.
So in theory, but in practicality, and then there's the cost. The success rate for those was really, really low. So it's not a practical option, in my opinion.
It's a possible option.
That may be a good area for additional development, to take it from the nascent stage that it's at now to something that would be a practical tool. Yeah.
Okay, so John Liu and I led the group that discussed education and training, and so I'll try to just give a few highlights of our discussion. We started out, Penny Riggs pointed us toward the American Society of Animal Science's Grand Challenge document, where they posed some questions related to what kind of training needs to be done, and so I think we'll get a hold of that document, and that could help inform our final product. In terms of our discussion then, we spent quite a bit of time talking about the need for interdisciplinary curricula and ways that we might go about pulling that together. A big emphasis in that discussion was on data science, and we didn't try to define data science, we just kind of used that as our term to know that we need to incorporate data-driven interpretations and analyses and so forth into our curriculum programs. We talked in that realm also about how to recruit students into animal agriculture training, either at the undergrad level or at the graduate level. And so we talked about the potential of bringing in, especially at the graduate level, students who have backgrounds and interests in data science areas like computer science or statistics, who may not necessarily have an animal science background. Engineering was another area that came up as we talk about phenotyping and sensors and things like that for future phenotyping. And then we also talked about kind of the opposite of how do we get animal science students who don't, especially undergraduates, who don't really have a concept of what the opportunities are, to understand the opportunities and then to prepare them for the job opportunities. And then one thing that came up was that sometimes when, especially graduate students, are good data people and programmers, that they don't necessarily stay in animal agriculture in their futures. They may migrate to other areas where they can apply their skills, and so we might need to consider ways that make the many job openings in animal genomics and animal agriculture attractive. One thing that we didn't discuss in our group, but Jerry kind of prompted me, and I'm going to stick it in here, is we didn't really talk about the funding mechanisms for grad students. So while there's a need for people in the industry, we also have a lot of challenges. I think we could train a lot more graduate students if we had the funding mechanisms in place to pay for those students. So that might be something for the future, to try to figure out how to address that disconnect of needing grant money to pay the students, but there are plenty of job opportunities for the students if we could put them through the system. And then another area that we spent quite a bit of time talking about was finding ways to partner with industry, and so in terms of helping us to create innovative educational models and to expose students to the opportunities that are out there. Another area that we talked about that we kept kind of coming back to was the area of science communication from the perspective of extension educators as well as communicating with policymakers and the general public, and so considering the different audiences that are out there and making sure that we and our students are trained to communicate with those audiences as well as to critically evaluate information that students that are the plethora of information that's out there that students find. And then another thing that we talked about was there are a lot of resources already out there. People do different things and different kind of training things, and maybe having a way to come up with some sort of repository or a location where training resources, training opportunities, maybe training methods could be kind of collected and made available to people. And so we talked briefly about, maybe there's something with the Ag Data Commons we could use, or something with the NAGRP site, that we could find a way to bring some of those resources together and make people aware of what other people are doing. So I think, in a nutshell, that was kind of the highlights, unless someone else in the group has something else. I know we kept kind of focusing on the need to get data science type things integrated.
I think also wanted to remember, I think we also talked about current career people as well as students in terms of providing those kind of resources that are out there and possibly facilitating exchanges between specialities, expertise, so that we can get cross-trained. And I think one thing we didn't mention, but just hearing the recap, we talk about needing people to be highly motivated to take on the additional skill and the different area of expertise. I think that people that are currently working probably are very highly motivated to learn what they need to do the science. Now that you're in the job, you know what you're missing. So that may be another way to fund that, rather than always just thinking of people who are current students as well. I think that was all.
So I was at a university industry consortium meeting many years ago in St. Louis, Archie. You may have been there. And my recollection is Monsanto was the host of that meeting, and they had 10 to 20 students that they funded on an ongoing basis with no commitment to returning to Monsanto as an employer, but they managed to recruit many of them. And I'm wondering whether we can reach out to industry partners and use FAR as a matching partner to put together some consortia to fund undergraduate or graduate students in whatever domain we're thinking is most important.
And just one other thing that we had mentioned too, I think, was the idea that the necessarily non-agricultural schools, especially where they have computer and bioinformatics courses, they're focusing on, usually human medicine, but that might be a place where if you're looking for a graduate student to work in your lab, a lot of times they're hungry for projects, so there may be a way to facilitate, getting a data science but biology person, but getting them to agriculture.
I would just like to add to Core's comment that the industry, but we should see the European model on that. They have a lot of mobility among student researcher. They have a program that has been working for years. So that's a model that works, is sustainable on time. Recently, I received a French student in computer science, and she helped me with some stuff. For me, it was a great experience, but it was short time because I had to split her time with friends, and I regret that. Maybe I will keep it her for the whole time, will accomplish more, but it was almost free for us and that opportunity, but she have funding to do that. So she do his work, she do his report, she learn how to work with some kind of messy data, and she report back, we gain, she want to, and everybody's happy. And that, maybe a consortium will be the way to get funding that can provide, especially for young students. And I will add to that, to the point that the universities and administrators need to maybe change his way how to get people tenure. Because if professors are going to spend a lot of time in this kind of program, there's need to be a way to give credit for the time that's spent in that kind of project. So I have different way to think the tenure program that have the university today.
I think Tim has a comment.
Yeah, just to follow up on your comment, Kurt. So at FAR, we actually are working on a program that is very similar to what you described, which ideally will be an industry and FAR supported training program. We're at the moment tentatively referring to this as a training program in digital ag, but this would encompass bioinformatics and computer science and a lot of what we've been talking about at the meeting here. So that's something that is definitely in the works, and so I appreciate your suggestion. And if you have, we can talk further about that and who might be companies that might be interested in working together on that. I'm actually not the point of contact for that program, but I'm happy to help facilitate in any way I can.
So I have something to comment on this. So we had a collaborative agreement. We were funding a student at the Roslin Institute, and part of that student's PhD program included industry interaction. And he actually came into my lab and spent a month in my lab, and that gave him the opportunity to see what is it really like in industry, what are the different things that you have to consider that are different in academia. I think that was very worthwhile and made the student a much more rounded individual when he completed. Also, there are a few, I would say not enough, opportunities that are supported. Like the Poultry Science Association has some undergraduate fellowships where the idea is that the undergraduate students go to an industry setting and work at that place. They're not to do it's not a focus to do research, but it's a focus just to work there for 3-4 months, just so they get some hands-on, some idea of what it's like to work in an industry setting, because it's quite different. And I think those are really worthwhile projects. That's how you get students interested. That's how you get students to understand what the issues are, put a different perspective on it, different than what they're picking up in academia. My general observation is that most academic settings are gearing the students towards replacing the faculty. We have success if our students get a faculty position elsewhere, and maybe there just needs to be a little different focus on what is considered to be success, because getting students into the industry is, at all different levels, is also a good measure of success.
Okay, thank you. I think we should go ahead and move on to hear from our group with the genetically modification.
Okay, this is an endurance race. So we struggled to focus during most of our discussion, and it was for a variety of reasons that are worth sharing. So one of those reasons is because for most of the mammals and for chickens, the technologies are in place now to do essentially anything you can imagine. If you can write down the sequence on a sheet of paper, we can make it. Largest transgene I've worked with is 2.4 megabases. The smallest change I've made is 1 base. I mean, we can do almost anything you can think of. But what we did recognize is that those technologies are not yet available in the turkey. They're not yet available in fish, and so one of the things that we'd like to see as a short-term goal are efforts to expand those technologies to species that are not yet represented in that club, as well as the emerging cultured animal species, perhaps shrimp or others. The other reasons why we struggled to focus to a large degree is because of the interdependencies. So at the end of the day, we recognize that we need to know what are good genes, what are important genes, what are good versions of genes, good alleles, what are the regulatory elements that we need. We're dependent on lots of other groups. And so I think I would challenge everyone, independent of this summary, to try to think about how do you identify important genes that are beyond the sequence space of your species. So as someone that does genetic engineering, I don't care if I get the gene from a warthog or E. coli or a bull. And so when you're thinking about your species, I challenge you to do that, and that came up to some degree, and maybe we can think about that as a short-term goal, how to do more comparative genomics, how to think about agricultural phenotypes outside of agricultural species, right? Frogs, for example, live in the nastiest places on Earth, and they almost never get bacterial infections. Surely, if we understood that, we could use that in agriculture via genetic modification. Another reason why we struggled a little bit to focus is because of the applications. For the genetic engineers that sat around in the group, we've been doing this for a long time. I've been doing this since '89. This is the first promise I have ever heard that USDA is thinking about entertaining making a genetically engineered animal. Right? There are no competitive grants that have actually put those on the ground yet. Hopefully this is a great opportunity. When we think about that, though, we begin to think about the impact of the perception of public perception. And I do put it that way on purpose. I do a lot of layman talks. I am not convinced we have a public perception issue, but we do have issues in corporate boardrooms. We have issues in administrators and government positions. We have issues at regulating levels all over the place. We have issues amongst each other. So one of the things that I think we need to get at is some of the sociology aspects, part of the educating of the public, what is biotechnology, what is a gene, et cetera. And then the other thing that we began to identify is because of that controversy and because of the mixed feelings about what will be accepted and what's not, perhaps we should focus on those things that I think we all can agree we could use genetic engineering for in livestock. And those would be validations of causative mutations, for example. Try and develop reagents such as one of the things that came up was lineage marked animals so that all cells of the immune system are red and you can flow sort them out pretty easily to do your transcriptome analysis. Or all hepatocytes are marked, or pick your favorite cell type and begin to build reagents that other people could use. But likewise, I think it's going to be very valuable, and we need to try to find a way to answer any scientific questions that apply to animal agriculture. What does this gene do? There's still a role of knockouts to look at the specific functions within our agricultural species, as opposed to that anomaly masquerading as a mammal known as the mouse. You know, we need to know this stuff in our species sometimes. And so the last thing that I'm going to concentrate on here is perhaps there is a focus that we should apply to trying to build resources that scientists that don't do genome modification could make use of, where you could begin to think, I want my gene knocked out. I think I found the causative mutation, am I right? You make a phone call, we make it for you at no cost and go back, because that cost is covered through some other plan with USDA. We have a similar facility in place already for swine and biomedicine that NIH pays for. We could think about a virtual model perhaps that's not tied to any one location. It's just tied to engineers that you rotate through. But perhaps that's a resource that we could start putting together. And I think I've been here long enough that if you guys don't have any question, we should move on to the next one.
Okay.
Well, there's a question in the back.
Two quick comments. One, funny you should say there's no transgenic fish
the only approved transgenic fish for consumption is Atlantic salmon, the only approved animal. And then there are a couple of labs. I mean, Morris sitting here was part of 1 or 2 proposals of using CRISPRs in fish, and there is proof of concept in a couple of fish species, rainbow trout, salmon, and maybe others that I'm not aware of.
Yeah, so I absolutely agree. What we don't have yet is the ability to make essentially any modification you can think of in a willy-nilly sort of fashion in the fish. But CRISPRs work, random integration works, but not in all the fish.
Okay, I think we got all of the groups. Is there a group that we missed? Okay, let's take a 10-minute break, and then we'll have our presentation from the National Ag Library.